Okay, so we're a couple of minutes in now, so I'll get started. Thank you all for joining on this very, very sunny day when I'm sure lots of people could be outside enjoying the weather. So thank you for joining us for this webinar. So this one is Transforming Healthcare Genomic Research and What It Means for You. My name is Isabel. I'm the Communications Lead at Central and South Genomics. We run these webinars on pretty much a monthly basis on a range of different topics, and today is Red for Research Day, as I'm sure many of you are aware. So we're trying to shine a light on research in genomics, the significance of it, and what it means for healthcare professionals, but also crucially patients and the patients that you treat and will treat in the future. So I'm just going to take over to housekeeping. So if you can please keep your cameras and mics off, just to enable our presenters to be able to sort of speak without interruption. We do one Q&A at the end of the session as opposed to sort of stopping after every speaker. So if you either note your questions down, you can drop them in the chat. The Q&A function is also open for this one. I'm not 100% on working that yet, but we will give it a go today. And our agenda is as follows. So up until about sort of 5-10 past, we've got our welcome and housekeeping. From 10 past 12 till 12:30, we've got Professor Diana Borrell. From 12:30 till 10 to 1, we've got Professor Sarah Ennis and Gora Cheng from her team. From 10 to 1 till about 1:05, we've got Rachel Podifat, who's going to speak about the generation study. And from 1:05 till about 1:30, we've got the Airlock team from Genomics England, led by Peter. And then we've got about half an hour for Q&A, as long as we stick to time as much as is possible. So we've got quite a few people joined now. So without further ado, I'm going to hand over to Diana, who I think wants to share your slides.
If that's all right, yes. Thank you, Isabel, and thank you, everybody. I have a slight clash with the question Q&A at the end. Shall I tell you why? Because there's a big BRC meeting. And that I think I need to be there by about 1:30. So I was just thinking then, if there's question and answers in the chat, I could try and answer them through the chat.
That's fine.
That's all right. And I do apologise.
Not at all. We have a little bit of flex in the timing. So if for just Diana, we can do the Q&A immediately afterwards, that's not a problem at all. And then everyone else will do at the end. No issues. And if there are any further questions after you've left, I can send them over to you by email.
I can look on the chat. Thank you. And thank you for having me. I just have a few roles. I know it looks silly, that great big long list, but in case you needed me for anything, these are the different things that I'm doing at the moment. So we all know this problem and the health economics of this problem, that rare disease patients go through this diagnostic odyssey, receiving many appointments, waiting many years before receiving their diagnosis. And I'm going to go through these first slides quite quickly because the main problem, we're doing great things. There's noise somewhere. I don't know if we can mute that person. The main problem is that we're doing really well at the moment with genomes, but we are missing mutations. I would say part of that is that our variant interpretation pipelines are highly protein centric, and hopefully I'll show you that that's not the only thing we need sequence for. Because very simply, our genes are made up of exons and introns. We need to cut those introns out, join the exons together, then that gets read, translated into a protein. It looks simple like this. I like this when I'm explaining it to people that don't understand splicing very much. It's like cutting up your DNA and making either a single-breasted suit or a double-breasted suit. So you make different RNAs from one DNA depending on how you cut and join it together. And it's a complex process with many factors involved. At its simplest, you've got a 5' splice site where there's the U1 binding, then there's the 3' splice site with various other factors binding. But people think it's just splice sites. Actually, there are factors really important in how exons are cut and joined together in the exon and in the intron as well. So that, for example, where the red splodge is, if there was a sequence change there that stopped that enhancer molecule binding, then that would also be a mutation. But that wouldn't be looked for if you're just thinking about how that sequence is read for protein. This is just to show you how very, very complex it is. And it's not just static, it's a three-dimensional process, all interacting together. So many years ago, we set out, we had been publishing before that mutations affecting the splicing macromolecule would cause disease. But there were sort of individual reports. So we set out to just show how many there were around and that they were a good cause of mutations. And we put a call out to any genetics department in the country. Have you got any variants of unknown significance, coding or non-coding, but that you think could be the cause because the phenotype fits? And I didn't need any bioinformatics or anything doing. And hundreds poured in. We now have over 800, I think. But the results on this first batch really showed how important it was to do this, because we did RT-PCR, RNA sequencing, and some bioinformatics sort of splice tools on it as well. And all the samples were collected in PaxGene tubes. And our pickup rate was 33% here. So we found abnormal splicing in 33% of the cases. All sorts was happening, with alternative splice donors, alternative splice acceptors, complex multi-exon problems, and intron retention. So that was the first pass where we really showed that we really need to think about this. At that time, we also looked at the tools that people were using in the diagnostic labs, MaxSense Scan, Splice Site Finder, etc., and Splice AI. And of those variants, really what you want is the chart to go straight up to 1 and across for it to be accurate. And you can see that they're all sitting at around 80%. Splice AI was the best, most accurate at 91%. But of course, that's 9% that it was getting wrong. So we still cannot totally rely on these tools. We do need a functional test. So just to give you some examples, here's a +3 in neurofibromatosis type 1. You can see it's skipping exon 10. You can see with these loops that exon 10 is now no longer included in the transcript. And why is that? Probably because it's interrupting the sequence where the U1 molecule has to bind for that exon to be included. In this case, SF3B4. This is an example of a more complex one with an alternative donor, alternative acceptor happening. And this one I've put in because it is deep intronic, really. It's not even near the splice site in P3H1. Again, very complex outcomes here with an alternative donor, alternative acceptor, and intron retention. So there were things happening that we couldn't necessarily have predicted, but importantly were causing the disorder. We even found new disease genes during this time. This was a family I saw in clinic, a consanguineous Romani family, 2 sisters with severe developmental delay and microcephaly. This was at the time when we were doing exomes. This exome was done in Exeter, and they were really clever because they said, we can see something, a non-tiered homozygous VUS in COPB1, which is not a disease gene, known disease gene, but it looked like a really important gene. So what could I do? Well, it was a +1, so a quick RT-PCR, and because not all +1s affect splicing, and the gel did show that it was affecting splicing. But what other evidence could we find? Near us in Portsmouth is the European Xenopus facility, and these girls had learning difficulties, small head and cataract, and they generated a tadpole with a small head and a cataract. So they really did show us that they had an animal model that matched our human model. And there's been a few like that that have come along. And then we thought, well, what about the ones that we don't have a VUS in? There's nothing picked up on the genome scan, but we really think that we could see if we can find something just with RNA, using the same sort of panels that we would use if we were doing whole genome sequencing. And we did do that. Carolina led this work, and in this group of cases, we found 4 new diagnoses, which was a 7% uplift in undiagnosed cases. So it is more difficult because you don't know exactly where you're looking, but it's still possible to find mutations not picked up with DNA alone. And I just wanted to highlight, we even see other things using RNA-seq, for example, skewed inactivation in this cohort, one case in this cohort. So examples here. So for those that aren't used to looking at these sashimi plots, the blue and green, the lumps are the exons, and the loops are connecting one exon to another. So the blue and green lines are the controls. And you can see the red in the patient, the loops are completely different to the control. So they're connecting different exons doing something else. And this patient had hypotonia, developmental delay, and the RNA-seq showed an intronic variant quite deep in the intron, 500 bases in, that was creating a cryptic site, and in a gene called AP4E1 linked to autosomal recessive spastic paraplegia. So that might fit, but of course this disorder does require a second hit. So Carolina went deeper back into the RNA-seq data to see if she can find a second hit, and she found a deletion which had not been picked up, not reported on the exomes. So that was really interesting. This is another case with profound developmental delay and seizures, microcephaly, hypomyelination on the MRI, and here she picked up a cryptic 5' splice site in exon 13 causing intron retention. You can see the green here at the bottom. The case has got lots of intron retention there. Oh. And we're not the only ones doing it. I just want to flash through these. There's many other people doing it, often in a particular disorder or in a group of cases. So muscle disorder, they actually did it on muscle samples. We were doing all this on blood because it's the easiest sample to take for us. So, but nobody had done it really in a very large way. So that's when we partnered with Genomics England, and I was involved right at the beginning with taking RNA samples for RNA-seq in the future, which was a great thing to do. And we didn't get 100,000, but I think there were about 25,000 or so samples. And so we undertook RNA sequencing with Illumina, short read sequencing with RNA and globin depletion on about nearly 6,000 samples, 5,500 or so. All the cases had no diagnosis from whole genome sequencing. And for those that are interested, on the left are the parameters that we used. We did quite a deep read, 100 million 100 base pair paired end reads. And these are some of the tools that we could use to look at the RNA-seq to find diagnoses. The main ones, though, that have been used are Outrider and Fraser too, actually. The demographics of the cases, 35% of them had neurodevelopmental disorders, 80% were white British, and that does reflect the cohort generally in gel. And the sequencing depth was very good. Question I get asked a lot is how many genes can we actually look at in blood? It's not perfect because not all genes, as we know, are expressed in blood, but if we can even get a majority, then that's great. So here, this is, I think, one of Jenny's graphs. So of all genes, 50% in the brown column, if the TPM means transcripts per million, and if that's over 5, then you can be pretty sure you can see something in the blood. So of all genes, 50% are expressed in the blood, but for the intellectual disability genes, over 70% are expressed in blood, and all the panel app genes, nearly 60%. So we're not getting everything, but it's pretty good. So just a couple of numbers. So with the Outrider, looking at expression outliers, you can see we found an outlier event in a relevant gene in 8% of the cohort, 8.3%. And here's an example. This is an RPL5 expression outlier, which is known to cause autosomal dominant diamond black fan anemia type 6, and that does fit the phenotype. And here's a splicing outlier using Fraser 2 in particular, which we found incredibly useful. And here we were seeing a splicing event in 10.5% of the cohort. So here's a splicing outlier example in the P10 gene. So blue and green lines are controls. And again, in the red, you can see the loops are different. And in fact, in the sequence, you can see an alternative 3' splice site being used. And again, the phenotype would fit with the gene. I think that's okay. So we haven't got an exact number of what the uplift in diagnostic rate would be. But even if we took half of those outliers as being relevant, then you would still estimate a pretty good uplift of nearly 20%, which is huge in that number of people. Other strategies were also involved, so I do want to shout out to Alistair and Damien Smedley and Letizia, because they really integrated their thinking into looking at these cases. So out of the cohort, we found outliers, 78 outliers. But if you integrated with structural variant calling, then we found another 19. And integrating with Exomizer, there was another huge batch. They did an absolutely amazing job. And this is now submitted, but out in preprint also. Another thing we did with that data was really try to find the second hit in recessive disorders. It was quite hard, but this is also published. Another potential use for RNA-seq. And you might all know Nikki Whiffin, who and her PhD, who found this also involving this data, the Renew syndrome, which was a newly identified syndromic neurodevelopmental gene in a non-coding gene, really, that was found by really thinking differently. And this has absolutely bloomed because it was thought to explain 0.5% of individuals, and now many other papers have come out looking at the other RNU genes. So RNU, do you remember I told you about the splicing process? So this is the U4 in the spliceosome, and mutations in that cause this disorder. So one of the things that we wanted to look at was whether short read sequencing was enough. Is there something else we could do with long read sequencing and catch other mutations? And we looked at both Nanopore and PacBio. Not many samples, but it has been enough to give us an idea. Really here with long read, you can detect novel transcripts. Really good at seeing intron retention. It gives you more resolution on the complex regions, and quantification, which we weren't great at doing before, and phasing of our variants. So these are the first 10 we did, and here on the right you can see in the green the cases where long reads did really well, and in the red where they weren't so good. But if I do it with some examples, for PacBio, here at the +3 UBR4 case, there was intron retention. It's quite easy to visualize and to see in PacBio. This one I wanted to show because here the RT-PCR was actually normal. The short read, this is the short read. So again, red is our case, blue and green are our controls. Shows a potential intron retention with an outlier, but you can see in the controls there's also some looping, so it's not really clear cut. But when you look at the PacBio, you can see the number of transcripts is many more in the proband than in the two controls of the abnormal transcripts, and you can quantify it. So our biosample 4 at the bottom is our case. The other 3 samples are controls. And you can see that in the case, there was 40 times as much abnormal transcript. And so we were unable to see that with short read, but we could see it really easily with PacBio. And Caroline has also published that. And just to highlight, this really is very good for intron retention and complex cases. So we, in 2023, got some money for a GMS transformation project to do this in a proper NHS system in the lab. It wasn't much, but we did do a few, I think it was 120 cases with the money from the GMS. Illumina also helped us with, I think it's another 150, 160 as well. So putting all those together, we now had 320 cases, all actioned in the same way, short read sequencing, RNA-seq, really trying to produce evidence as to why we need this in the diagnostic lab and how to produce guidance to really try and embed it. Same, a call went out, we were inundated with samples. We were happy to take VUSs, no VUSs, or trying to see if we could find the second hit for an autosomal recessive case. And these were all sequenced in the Birmingham Women's and Children's. So just to summarise, here on the left in panel B, the results, we had all sorts of results. Intronic variants causing mutations, synonymous variants causing mutations, missense variants causing mutations affecting splicing. So we must keep in mind that these variants have these names, missense, but it's not anything to do with that. It could be because it's affecting splicing. Looking at Splice AI again on the right-hand panel in C panel, if Splice AI was over 0.2, then we had a good number of positive results, positive mutation results. But even if Splice AI was less than 0.2, we had some results. So that's the 9–10% error that I spoke about before. Sorry, this is a bit small, but if we start at the top panel, this is in the guidance that or how we did it, and which developed into guidance post this transformation project. We had 2 routes to come into the project. One was if Splice AI was greater than 0.2, or the clinical route where a clinician says, actually, look, I've got this patient. I'm pretty sure this VUS would fit this patient's phenotype, but I can't prove it. If the TPM was over 5, then we would go straight to RNA-seq. If the TPM was between 1 and 5, then they got RT-PCR through the R296 route. And if the TPM was lower, then we would try probably to find another tissue that it was expressed in. Our colleagues, our diagnostic colleagues in the Netherlands, for example, tend to just use fibroblast skin biopsy for RNA-seq. They say their yield is absolutely excellent. Okay, so actually in this cohort, we had a 55% pickup rate. Of course, it was a biased cohort. They were the top tier that people wanted to put forward, and even a few in the recessive cases. So I'm coming to the end because here I've just presented to you lots of evidence for making new diagnoses and how we could do that. I have put in a test directory. We've worked with the GMS to put in the genomics unit, in fact, to put in a test directory application. It did go in quite a few months ago. I haven't heard, so it's not live, but I have worked really hard to try and do that. But all these cases are interesting to people also because you can potentially correct that abnormal splicing. through RNA-targeted therapies, the most famous of which is Spinraza for spinal muscular atrophy. So this really, this is a field that is growing hugely. And I think it's, this slide is well out of date, but there are many antisense oligo drugs authorized to date. And Andrew did write a very nice review in 2021. We've been working on that too. So just briefly, we're trying to group the splice mutations to try and see if we could develop antisense oligonucleotides for each grouping. And the other thing we're looking at are small molecules, even pre-existing drugs that change the splicing and change the transcript levels to a more optimal transcript and not so much of the abnormal one. And we've been doing that in some of the cardiac genes really well. So there's so many people to acknowledge. One day I'm going to really write down all the doctors' names that have been referring and recruiting, because it has been amazing. Thank you very much to the Birmingham labs and the Salisbury labs, because they've been so helpful in all these projects. Genomics England, Illumina, Nanopore and PacBio have all contributed. And the NIHR and the main workers are there in orange, who have really done most of the thinking and the lab work, and Genomics England. Thank you.
Thank you so much, Diana. It was really interesting. So we've got one question in the chat. We've had a few throughout, but they've mainly been answered. But we've got one question from Imogen, which is, at the moment, what kind of RNA testing can be requested by clinicians, or how much is RNA testing utilised in the NHS? How do you see this service developing in the future?
Yeah, so I know that only certain GMSs do RNA testing. It does exist on the on the test directory as R296, but not all centres are doing it. So that would be an RT-PCR. We have the test directory sort of application that's gone in is RNA sequencing, a high throughput way of doing it. So you don't have to, it's not bespoke for each mutation. And that would be the ideal. At the moment, it's only for variants of unknown significance, but it's not live, I'm afraid.
Thank you, Diana. Are there any further questions for Diana at this point? If any come to you later, you can send them via email and we'll get them over.
Of course, thank you.
Okay, so thank you so much for that presentation. I'm sure everyone found it as interesting as I did to hear about the ongoing work there and the outcomes. Sarah is now presenting. So we've got Professor Sarah Ennis with us, who's a professor in genomic medicine, a consultant in clinical genetics, a co-director of the designate BRC Southampton, and a genotype phenotype theme lead for Genomics England, as well as having a role within as a research director at Central and South Genomics, and I'm sure others as well. So I will hand over to Sarah and we'll hear about her work in this space.
Lovely. Thanks very much, Isabel. Can I just check, are you able to see a little pointer on your screen at the moment?
We are, yes, I can see that.
You are, good,
yes.
Good. That's great. Lovely. So I'm going to talk to you about some scalable methods to, oops, I'll go back to that one. Some scalable methods to help prioritize genes for genomic diagnostic and for research uses of medical genomic data. So I'm going to start by talking through some of the problem really that is rare variation. So you've heard Diana talk lots about variants of uncertain significance. If you have a variant that's common in a population, like, for example, the delta F508 in the cystic fibrosis transmembrane receptor gene, well, then it's well established that that variant is pathogenic, and it occurs in less than 5%, but more than 1% of Northwest Europeans in the population. So it means it's a variant of known significance, not of VUS at all. and that can be quite good. And very often, if a variant is very common in a population, for example, half of the people on this call carry the variant, well, then the chances are that that variant is benign. And we often do make that assumption when trying to filter variants down. If they're very common in the population, they're very much less likely to be causing disease. So when we're analysing huge amounts of genomic variant data, what we have to focus on when we're trying to interpret variants is these rare variants, which tend to more often be variants of uncertain significance. And the thing about these rare variants, there's been some studies in some large cohorts looking at, and one of them was 1,000 Genomes Project, and they identified 88 million single nucleotide or variants across a fairly modest cohort of just 2,500 individuals. About 85,000 of those were just variants at a single nucleotide, and there were lots more short indels, about 3.5 million of those, and about 60,000 structural variants. And if we took any individual on this call and we sequenced your whole genome, we would see that any individual has about between 40,000 and 200,000 variants that occur in fewer than 1 in 200 people. So they're quite rare. And many of those variants actually they truncate proteins. So they change the protein quite drastically. And then another 10 to 12,000 of them alter the amino acid sequence. So that's in any normal, healthy individual. So as always with genomic data, we get an abundant amount of data, and it's always trying to find the golden needle in a haystack of needles. Trying to find the single variant or a handful of variants that might be really driving disease in individuals. And this gets even worse, this problem of trying to look at these rare variants, it gets even worse when you look at across populations. So if you looked at this group of 2,500 people from 100,000 genomes, and you looked at the number of variants in this graph down here, the number of variants on the y-axis that have a low minor allele frequency. So an alternative allele frequency of about 0.5 means half of the people in the population have the reference allele and the other half of the people in the population have the alternative allele. So again, these variants are really unlikely to be causing disease. But what we see is when you get down to the very rare variants, their frequency is enormous collectively across large populations, such as the Genomics England population or the population that we're now referring into the Genomic Medicine Service. The more and more people we sequence every day, and we refer for sequencing in the Genomic Medicine Service, the more data or the more variation we accrue that are, one, very, very rare, and two, that means we're much, much less likely to know what they do, because there won't have been these sophisticated laboratory tests that Diana has talked about, looking at individual splice variants and trying to actually confirm the functional relevance of all those very, very many abundant, very rare variants. But the problem is, although most genetic variants that affect human health are very rare, we have such an enormous amount of them to look at when we're looking at populations and trying to serve the population across England in the NHS Genomic Medicine Service. So this just shows actual data for just one gene. This is the breast cancer 1 gene, BRCA1. And again, on the y-axis, we've got the minor allele frequency. So how often we see a different allele, a different A, T, G, or C at a position across this gene. The blue bits at the bottom indicate the coding regions of the gene. And what you can see is if you look up here about at 0.5, so that's when half the people in the population have the reference allele, and the other half of the population have some alternative allele. And you can see even down to here at 1%, there are large numbers of variants that occur in only 1 out of 100 people all the way across the BRCA1 gene. But the real problem is down here. When you get to the very, very rare variants, you can see that their frequency across the gene, it's almost a solid line. Because when we sequence more and more and more people, and these data are actually from gnomAD as opposed to the 1,000 Genomes Project, and gnomAD collects hundreds of thousands of exome and whole genome data. But you can see the frequency of incredibly rare variants across the BRCA1 gene is just enormous. And these rare variants are the ones that are much more likely to be causing disease, but they're so abundant, they're very hard to statistically analyze. So if we come and try and, for example, use traditional association tests, and we take any one of these very, very rare variants that we find in the cohort, and we ask, well, who has this variant and who doesn't? Well, the vast majority of people do not have that variant. So if you're looking mathematically at a grid of who has which variants, you end up with this sort of very, very sparse matrix. Most people do not carry most of the variants. So it's a very, very sparse data set when we look across very big cohorts, and it's filled with these rare variants that are much more likely to be causing disease. Now, this is a busy slide, but this is, I'll just walk you through the top. This is a solution we came up with to try and, instead of modeling each one of those rare variants one at a time and creating this very sparse matrix, collapsing the set of variants that are found in any one individual into a score that I can compare my score against Isabel's score, against Diana's score, against anybody else's score on this call, and say, well, I have a high burden of nasty variation caused by whichever genes happen to be personal or private to me or rare in my genome. And how does that compare to the loading, the pathogenic loading in somebody else? And the higher the score, the more carriage of nasty pathogenic variation. And so what this model does, or this algorithm called genomic does, it's a gene level pathogenic burden scoring for individuals. So we're no longer looking at the variant, we're looking at a score for an individual gene. And it takes in the VCF file, so the variant call file. So that's the list of all variants. If you've been whole exome or whole genome sequenced, it generates a VCF file for every individual, which is the list of and the location and the nature of the change of every variant that any one individual has across their exome or their genome. And then we identify the variants in one gene. So this could be the ABCD1 gene, it could be the COPB1 gene that Diana just talked about, it could be BRCA1, it could be any gene you like. And it takes in that variant call file, and it focuses on, well, how many, if this is individual X, how many variants does individual X have in the ABCD1 gene? And it takes each of those variants, and in turn, it annotates them with information. It asks, well, what is the allele frequency of this variant 1 in the population? What is its deleteriousness? So there are various deleteriousness metrics out there. We happen to use CAD quite a lot at the moment for reasons I can explain later on. We're not married to it. If a better one comes out, we will change. But we find it very good because it scores all variant types, not just coding variants or missense variants. And of course, we're interested in all variants, including those deep intronic ones and the ones in between genes, perhaps sometimes as well. And of course, we don't have, for most genes, we don't have just one copy. We have two copies. We have the one we inherited from our mum and our dad. So we record the zygosity. And we put these 3 bits of data into the genomic algorithm, which gives a higher weight to very low frequency variants, and it also gives a higher weight to any variants predicted to be nasty, and it accounts for the heterozygosity, or sorry, the zygosity in that individual. And we then we create a variant score. And then we take those variant scores for variant 1, variant 2, variant 3 in individual X, and we sum them. So we end up with one score for individual X for this gene ABCD1. So individual 1 here has a score of 7.7, and we do the same for individual 2, individual 3, down across our cohort, and we do that for all genes. So we end up with a matrix of all genes in the genome. So no matter how many people we sequence, if we sequenced everybody on the planet, I couldn't even guesstimate the number of variants, many billions of variants theoretically we could observe. But in actual fact, this matrix is limited to the number of genes in the genome. So we'll get in excess of 20,000 genes, and then there'll be a row for every one individual. And we will have a score whereby, for example, here for the ABCD1 gene, we can see that actually individual N and individual 1 have high scores for this gene. And in the Titan gene over here, individual 3 had had a very high score. Individual 1 also had a high score. So you can compare and see who has the highest loading of nasty variants. And then you can use this matrix to compare the scores across individuals for any gene. So it may be that you have a particular interest in SCN1A, and you can take out all the scores for those that gene, and you can then reverse the paradigm. You can go from the gene and ask about the phenotype and say, well, I've got all these people with nasty scores in this gene. Do the people with the very nasty scores, do they have a phenotype in common? So it's reversing the paradigm of how we usually have found disease genes. We usually start with, okay, I've got 20 people in a family with epilepsy, or I have 100 people across this cohort with epilepsy. Can we go and find a genetic signal? This allows you to say, I've got a bunch of people who have a really high score in this gene. Are they enriched for any phenotype? So it switches that paradigm. But it is also possible to just look at an individual and say, for example, individual 3 here, and you could ask, well, individual 3, do they have extreme scores compared to everybody else? Do they have extreme scores in any one gene in the genome? And it may well be that they have extreme scores in just one gene. And you can then ask, well, does that gene map to the phenotype of that individual? And that's another approach that we've used in some of the slides that I'll be bringing up in a couple of minutes. So this is really just an alternative way to collapse the vast amount of genetic data you observe and genetic changes you observe in any one individual or patient, and collapse them to a score that maps to each gene. And you can then look at any individual's score across their genome and say, relative to everybody else in a reference population, or a group of patients who do not have that disease, where is this individual remarkably outlying? And we have done this. So this is Guo and Iman from my group. So Guo helps maintain the pipeline, and Iman helps make the pipeline work efficiently, because when you're actually processing data over such large data sets, a whole genome is enormous. And then when you have a whole genome for tens or hundreds of thousands of patients, you need to make your code very efficient. So we tend to write our code in modular components and wrap them up in Nextflow wrappers. And that means then we Dockerize them. So it means that somebody else could download the image, and it's sort of, you open up the package and everything else, everything you need to run the software to get the sort of same results is all in one Docker image. But of course, we've had to do that in multiple places. We've done it on so Amazon Web Services. That's the platform that's used by Genomics England and UK Biobank. UK Biobank's not quite accessible at the moment, but we are working very busily in Genomics England, and we've created this matrix on the aggregate 2 data. And the aggregate 2 data is basically the 78,000 whole genomes that were done on germlines as part of the 100,000 Genomes Project. And there is actually an aggregate 3 data set has become available this year, and that's about almost 140,000 whole genomes, all stored within Genomics England. And in that set, it's got all the 100,000 genomes, but it's also got some individuals who weren't aligned to the same version of the human genome as part of the 100,000 genomes. And it's also got some COVID samples, about 20,000 COVID samples. And very interestingly, it's got about 30-odd thousand of the Genomic Medicine Service patient data in there. So some quite new data to be looking at in there. Iridis is our local supercomputer in Southampton, and we've deployed the same GenP algorithm on a bunch of patients who actually don't usually get referred to the Genomic Medicine Service. These are patients with inflammatory bowel disease, and I'll show you some results with that. So our pilot project looking at GenP and gel was with this very talented lady. She's studied studying to be a pediatrician, but she was doing her PhD group in my group at the time. And we deployed Genepi on just a select bunch of recessive disorders. And we picked up over 100 missed diagnoses by using this Genepi tool to identify signals that correlated with patients' symptoms. And we've now, as I say, redeployed this on all 78,000 individuals across all genes in Genomics England. And inside the Genomics England research environment, there's this special cloud area to do high performance compute, so called LifeBit. And we've got our Dockerized images working in there. And we've managed to make the matrix for the aggregate 2 data set. At the very beginning, when we were just sense checking the data, we went in and we looked at individuals. So this is individuals whereby we saw these 2 affected siblings. They presented with pediatric motor neuron disease, and we identified that of all the Genepi scores for this gene SCO2, which is a recessive gene known to cause pediatric motor neuron disease, these 2 individuals were ranked absolutely highest, or they were ranked equal number 1 in Genomics England for this gene. And this was a gene known to cause the disease they presented with. So it was a very rapid way. Once we had this signal from the gene, you can sort of go back and look at the individual variants, put them through the ACGS standard and NHS guidelines, and confirm the diagnosis. And this was confirmed as the diagnosis in these individuals. Again, it's completely agnostic, so you can run this regardless of panels. So, and we did run it regardless of panels initially, but obviously when it comes to the ACGS interpretation, it's very important that the genes we identify might be part of the green panel app for that referral, or at least we can go back and get additional clinical information and confirm. But this was another one where we had this family that was hugely affected by this cataracts, and we had data on the father and the son, and again we identified they were joint third for this CRYBB2 gene, and it ended up being, when we looked under the hood what was causing the high Genepi score, it was actually variants that caused a dominant known loss of function. So again, this was another diagnosis that was picked up, and there were many more. In obviously Genomics England is quite enriched for intellectual disability as a referral phenotype. There were lots more we were picking up, so we were confident that this was working to pick up diagnoses at scale. And Goa has now just completed this analysis, and what she did is she focused on about 2,500 undiagnosed probands. So these are people who had a negative report in the exit questionnaire. And when she identified in those 2,500 patients, arbitrarily, did any of them have a score that was ranked in the top 10 for any genes? And if they were, then they were put forward, those genes and those individuals were put forward for processing. And that processing was obviously according to the clinical guidelines. So this was, we were trying to uplift diagnoses as opposed to find new disease genes here. So we were restricting things to the green gene panel app, and we were adhering to all the rules, the ACGS guidelines for diagnosing a disease. And we are working through the results at the moment with the clinical research interface team at Genomics England. But so far, it looks like across 421 pedigrees, and of course, in some pedigrees, there's multiple people affected. So it's actually about 507 affected individuals, probands and affected individuals. And these all look to be pathogenic or likely pathogenic variants, all meeting the ACGS guidelines. So an uplift of about 500 diagnoses, and that paper is under publication now. And I just quickly wanted to go through an application to inflammatory bowel disease. Again, this is a disease, it's only children aged less than 2 who are diagnosed with IBD and have very severe symptoms. They're the only ones who are allowed to be referred right now for genomic medicine service testing. At the turn of this century, we really only knew about 1 gene in IBD. GWAS pinpointed in excess of 200 additional genes, but none of those were being used in the clinic. And we've integrated Genepi for the genetic signal with large language models to pick up discrete clinical phenotypes, because that's what you need when you're looking at a common disease. You need the detail of the specific phenotype rather than the umbrella term of Crohn's disease or psoriasis or rheumatoid rheumatoid arthritis. We want the detail of the phenotype that's very often hidden in unstructured data in the clinical record. And so the individuals in our local study have all been consented for research, and we've been able to access their unstructured data. And we use that a text input and a large language model, which we train, and then we get numeric representation of the output. In our hands, we're often trying to get that numeric representation is actually an HPO term, which many people in the genomic medicine service are familiar with. So we use these large language models to help scrutinize some of the absolutely abundant unstructured data that are in the electronic healthcare record, and that nobody has time to go through and read and code into HPO terms or ICD-10 codes. You can get a large language model to do this at scale. So we did this for the IBD cohort. We took the unstructured data that's written down in the histology and the imaging, so it could be MRI or CT scan. And for our cohort, we had 1,500 patients at the time this was done. There were 32,000 of these reports, so we can't get humans to read through those. We can, and we did, develop a large language model to do that, and to end up structuring the data and turning it, in our case, into HPO terms. And so this is, and we're actually in the process of doing this with clinical letters now. Of course, all the letters have to be redacted. The large language model goes through and finds the specific phenotypic manifestations in the individuals. It gets rid of typos and it turns them into code, and you end up with this list of HPO terms for your one patient on whom all those clinical letters were written. And when we do it on a pathology report, this is what an unstructured pathology report looks like. We turn it again into the pathology statements, and these statements again get turned into SNOMED CT. Uberon is a coded system for anatomical location, or a SNOMED CT, or in our case, we often wanted to get HPO terms because that's often the language of phenotypes used in the genomic medicine service. And this is one example for one of our local patients. Interesting, this is a young man now. He's aged 21 years, but he was diagnosed with Crohn's disease at age 11. We generated his GENOPY score amongst the cohort of IBD patients we have locally, and we normalized all the scores. And so instead of a score for Titan getting up to 22 and a score for a very conserved genes getting up to 8, we normalized all the scores for each gene between 0 and 1. So these are the scores for this patient. So way down here on the y-axis, these are all the genes whereby this individual had a low score compared to everybody else in the cohort. And these are the genes where the individual had a high score. And you can see that they have a reasonable number of genes where they have a high score. That in itself is not hugely valuable. When it becomes really valuable is when you add in the clinical phenotypes. So we extracted all the clinical phenotypes from the patient, and we looked how often, or what is the overlap between the phenotypes in the patient and the phenotypes that are known to be associated with the NTRN gene or the MMUT gene or the MTHFD1 gene. And we asked what was the overlap. And basically, if you have a high score in the x-axis, it means that the patient has lots of the phenotypes associated with STAT3, for example, but they don't have any genetic burden in STAT3. So they don't have any nasty mutation. They have lots of phenotypes overlapping with what's expected for CARD8, but again, they have a very low burden of nasty variation in CARD8. But what we see up here in the top right corner is where the individual has a high burden of variation, amongst the highest in the cohort, and their phenotype overlaps with what's expected for that CTLA4 gene. So this patient's diagnosis actually changed on the basis of these results. We sent some blood up to Great Ormond Street, and they were able to help confirm the functional impact of the variants that were causing this CTLA4 deficiency in this individual. And the diagnosis has changed from Crohn's disease to CTLA4 deficiency. And because of that change, this patient could now receive a drug called abatacept, which is curative for CTLA4 deficiency. And of course, they're young, they haven't yet had children, they know the cause of their disease, and that information can be used in future family planning. And we have similar examples across other genes, XIAP, L4, so all where a common disease diagnosis has now changed to a monogenic inborn error of immunology in many cases. So at the moment we're updating the GENOPY algorithm. We're actually applying it to the aggregate 3 dataset in Genomics England, and we're adding to our IBD cohort both the clinical and the genomic data, including other international common disease or inflammatory bowel disease patients, hoping to demonstrate evidence that in actual fact there's a not unreasonable, it will be a low number, but it's a not unreasonable number of patients who are somewhat burdened with a common disease label, because when you get a common disease label, it means you actually can't access these medical tests, the genomic test. And we're generating increasing evidence that in actual fact, sometimes a genomic test is something we should consider for people who present with severe symptoms of a common disease. So I'll finish up there, and I'm happy to take any questions.
Thank you so much, Sarah. That was really, really interesting. We've got a couple of questions in the chat. So we've got the first is, did you get different variant burden in different genes against different geographical regions where patients reside?
Yeah, I suppose one of the thinking about what's underneath your test, your question here. One thing we have done, we've looked at the GENOPY scores in about 200,000 individuals in UK Biobank. And so we have, and we looked at the G6PD gene, and that G6PD gene can be very heavily involved in malaria, and you end up getting different frequencies in different variants in individuals of different ethnicities, and more importantly, individuals who come from populations where malaria and malaria-bearing mosquitoes are endemic. And the answer is yes, you can see some population differences. And of course, older populations, so African populations, for example, have a much higher pathogenic or a higher variant burden than, for example, Northwest Europeans. But one of the ways we can control for that is actually limiting our GENOPY score to those variants that are nasty. So even though different ethnicities could get different scores for different genes, there is no reason why, for example, a different ethnicity might have a higher burden of disease-causing mutations. So there are ways you can control for that. And absolutely, things like olfactory receptor genes, they will accrue mutations, but they're not relevant to disease. So those are genes that are degrading because we're cooking and refrigerating our food at the moment. But other genes, like, for example, the IL-10 gene, that will have very, very little variation no matter what patient cohort you look at or what population cohort you look at, because it's very intolerant of variation, and it does more often lead to illness, for example, if you accrue nasty variants in it. I hope that answers the question.
Thank you. And just one more at the moment. So we've got, how do you quantify what a high burden mutational score is for further investigation?
Yeah, so that is the reason why you need a big reference cohort. So if I just created the GENOPY score from my own genome and I got a score of 3 in gene X, what does that mean? It means nothing. You need a reference population, and this is why we're trying. So we've made the GENOPY matrix for 200,000 of the UK Biobank cohort. We're now making it for about 140,000 inside Genomics England. So it means if there was a patient referred next month into the Genomic Medicine Service, and we generate their GENOPY score, you would compare it to the scores of against some reference data sets, such as the aggregate 340,000 individuals. And then you could say, well, is the score of 3 high in that cohort? And if there are many people with scores of 30, well, then you know it's not a high score. So you compare it against a reference population. And that's why we are making that large reference population inside Genomics England and inside UK Biobank and these various areas against which you can assess and benchmark, is the score you're seeing in this gene in your patient, is it high compared to that reference population or not?
Great, thank you so much. So thank you, Sarah. I think Sarah needs to jump off now, but if there are any further questions, much like with Diana, please send them over via email or pop them in the chat, and we'll do our best to get them over
Brilliant.
and answered over email. But thank you so much
Great.
for your time today, Sarah.
Thank you. Thank you for having me.
No problem. I'm now going to hand over, hopefully my screen will be sharing in a second, to Rachel Pudafat, who is the Regional Results Coordinator for the Generation Study in the Central and South region, which stretches from sort of Stoke-on-Trent down to around the Isle of Wight. And she's also a midwife sonographer at Birmingham Women's Hospital. So I'm just going to hopefully try and find the right slide. Rachel, do we have you on the call?
Yeah, can you hear me?
Yeah, I can hear you absolutely fine. I'm not sure if I'm able to give you control. So if you're happy to do next slide, that would be great.
Yeah, I've got
Perfect.
control of it. I think so.
Thank you. Okay, great. Thank you.
Yeah. Good afternoon, everyone. So just like Imogen said, I'm the Regional Results Coordinator for the Generation Study. So the Generation Study is formerly the Newborn Genomes Programme. It's a pioneering UK research initiative by Genomics England in partnership with the NHS. It's the first of its kind using umbilical cord blood from birth to sequence the genomes of 100,000, hopefully, newborn babies to detect over 200 plus rare treatable genetic conditions, hoping to give life-changing interventions. So there were three main aims of the Generation Study. The first was to evaluate if whole genome sequencing can effectively screen for more than 200 rare genetic conditions. And it was also to enable wider research to gather genomic and health data so that it could be accessed by universities, charities and pharmaceutical companies, which I'll talk to you about a bit later. And it was also just to see if it was worthwhile or beneficial to store someone's whole genome over the course of someone's lifetime. So at the Birmingham Women's Hospital, although I'm the Regional Results Coordinator for the Central and South, I'm based at the Birmingham Women's Hospital. So I cover the results for a lot of Birmingham hospitals, but I'm based here on recruiting as well. So we go to all the inpatient and outpatient areas, including the wards, antenatal clinics, the day assessment unit, glucose tolerance test clinics, obstetric waiting rooms where people come in for their caesarean section. So this is not an exhaustive list. There's lots of other places, but they're the main areas that we do go to. And we have a screening log as part of our site file where we register all self-referrals that come from people scanning a QR code that are on leaflets or pull-up banners and posters around the hospital. So our community midwife discussed the study at the 16-week check when pregnant people have their 16-week check with their community midwife. Also, our radiographers are meant to give out the patient information sheet provided by the study at the mid-trimester ultrasound scan. We have translated the document into 18 different languages, and they were meant to be the most common languages that people required. So it's not area specific. So we've got them in French and Spanish and things like that. And that might be more population dense to sort of like London area. But ours is mainly Urdu and Arabic, if I'm massively honest. We've got a really diverse population in Birmingham. It's our unique selling point for research. And we often use interpreters and interpreting devices. So face-to-face interpreters, and a Dora interpreting machine is effectively like an iPad on wheels, where you can dial up in real time an interpreter that they'll then translate to the patient in that language. And we try not to use relatives of people, which is not a hospital policy to do so. The eligibility criteria to join the Generation Study, you had to meet all of it. So you have to be over 16 years of age, which is most research projects. You have to be registered with a GP in England and hold an NHS number. You had to be planning on giving birth that are participating in NHS taking part in the Generation Study, and you had to be pregnant with just a singleton baby. Unfortunately, the portal, which I'm going to show you, would only accept one baby per pregnancy. So futuristically, they'll have to look at a way of getting around that. And you must not be acting as a surrogate or plan to give the baby for adoption, because they weren't going to give results out to people not doing the ongoing care of their child. So it does not replace the standard care that's currently in place. So newborn babies, they currently, on day 5, they are screened for 10 rare genetic conditions via the blood spot. And if you're a parent or if you've been through a hospital system, you'll know that this is the standard newborn blood spot test, where the community midwife comes out to your house on day 5, pricks the baby's heel, does 4 dots of blood on a blotting card, which goes over to the Birmingham Children's Hospital and screens for these 10 conditions on the right hand side, which HT1 was added in June last year. The generation study screens for 200, over 200, it's 248 actually, but sometimes they remove genes and add genes, rare genetic conditions from umbilical cord blood. If we were unable to get the umbilical cord blood, we can approach the parents and say, we were unable to do, and your baby can have a heel prick, but that's separate to the newborn blood spot test, and we make that really obvious to them. A list of all the conditions that we screen for can be followed on this little QR code that I've given. In order to meet the criteria to go into the generation study, they had to be treatable conditions and have a care pathway already within an NHS system. So this is just a little look at what the portal looks like that was created by Genomics England. So you just pop the person's NHS number in here and their date of birth, and it'll bring them up. So this is a dummy profile myself in here. I'm not childbearing age, just like to say. So basically it'll take you through the eligibility criteria first of all, so you can't go any further, so that you know that that person's going to meet the eligibility criteria. And then there are 11 points of consent that they must know, that they have to be happy with, that they know that it's optional and separate from their baby's standard NHS care, that their babies are the parents involved in their decision, which is probably one of the few research projects where we approach pregnant people, where we say your partner really should know about this, because if you do get a condition suspected result back, it may then lead on to further tests, and so we want him to be involved in that decision. If they're not in contact with the babies of the parent, they don't have to get permission to do that. So I'll just skip through that. But there is one part on here about the data and samples, and it says on here that the anonymised data could be accessed by charities, universities, and pharmaceutical companies. So they are aware of that. And also it tells them here that they're going to store it for 16 years, because when their child is 16, they'll get consenting rights. So then they'll be contacted by the NHS portal to ask, do they want to store that themselves, because they've got consenting rights, because it's their sample. And then in the portal, the useful things to know is that we just ask for ethnic origin, and then any sort of genetic ancestry. And most people will say, oh, I don't know of any genetic conditions in our family. And that's normal, because they are so rare. So most people would never know their grandparents' genetic ancestry sort of. And also we didn't know as much about genes then. So, you know, if you had relatives that in the far distant, you know, past that died of things, they didn't know, they couldn't pick it up. So I just always type anything when I'm asking them about it that could be of relevance. So most people in Birmingham say they've got a family history of diabetes, or if they mention that, you know, they've got a heart murmur, or someone's got high blood pressure in the family, because you don't know what conditions will be linked into the various different genetic conditions. So I find it's quite helpful to put those things in. So as of June, the 8th of June 2026, we have recruited almost 70,000 people throughout the country. That's not just here at this hospital, that's all the hospitals involved. And over 55,000 samples have been received for whole genome sequencing. And we've returned over 49,000 results. Now that is more now, but this was just the results as of June, and then there'll be another update in July. So the success stories, I would love to tell you about all of the success stories, but obviously this information is really confidential, and so I can't tell you about rare condition results. Unfortunately, I would love to tell you about all the success stories, because we have picked up a lot of results. But I can tell you about this one, little baby Freddie, because his parents were so amazed by the generation study. They wanted to speak out about it. They went to the press. They spoke to communications. They wanted their story published. So little baby Freddie was picked up on the generation study with the RB1 gene, which is associated with hereditary retinoblastoma, a rare type of eye cancer that affects young children. And he was referred to the Birmingham Children's Hospital, and they undergo a mix of chemotherapy and laser treatment, and which treated the tumours in his eyes. So, and this little QR code here will take you to the story that his parents told. So, what does the generation study hope to achieve in the long-term future? So, for babies like Freddie, it was obvious, wasn't it? We diagnosed him young. It wasn't picked up, you know, in the newborn test. He didn't obviously see those things. And now he has been referred into ophthalmology and will have like follow-up treatment from the clinician that he needed. But what about the babies that don't take part in the generation study? So all of those stored samples go into a biobank, and it's hoped that we can understand whether analysing babies' DNA can establish a new healthcare standard for screening and diagnosing and treating young children. So it came into the category of prevention rather than treatment in the NHS 10-year plan. And it's the first of its kind. So no other research has done whole genome sequencing from birth or from umbilical cord blood. It has been hoped that by 2035 that this could be integrated into NHS care. But the generation study are going to sort of help see if that's feasible and how the logistics of this are going to work. So it's hoped that that it can include a genomic that this inclusive genomic database will benefit all of the population and deepen our understanding of rare diseases. But if the samples in the biobank are not diverse enough, it might jeopardise our chances to understand how rare diseases can affect our wider population. So most of our recruiting sites, I think it's fair to say, have a larger amount of white British population, and I think that's true of quite a lot of UK studies that happen. You do get more sign up from this type of educated group of population. However, that's great, isn't it, for that group of population, but it doesn't benefit everyone if that's the only stored whole genome sequence in the biobank. So yeah, I don't really know. Obviously, we're still in the process of collecting the samples, and it's going to take years to analyse all this information. But let's see what happens in the future.
Thank you so much. That was really, really interesting. And it's great to hear about how the study is sort of embedded in sort of the real world of all of this research that we're doing. So we're going to go back to Q&A at the end, if that's all right with everybody. And so I'm going to hand over to Peter O'Donovan and Gina Staunton, if I have you both on the call.
Yeah.
Hello.
Hello.
Fantastic. Hi. So please feel free to overwrite my share. I think you should be able to share your screen. But we have with us Peter O'Donovan and Gina Staunton from the Airlock team at Genomics England. So obviously we're talking about all of this research, and all of it produces data. And then when that data goes live to researchers and to other places, as discussed with the generation study, for example, there is a sort of gap in between that of making sure that it's sufficiently pseudonymized, anonymized. Peter and Gina are the experts on this process. So I'm going to hand over to them.
Hopefully everyone can see those slides now.
Yeah, we can. Thank you.
Okay, great. Thanks very much. Thank you very much, everyone, for having us to talk. We'll be very happy to answer any questions you might have at the end when we've gone through the presentation. Myself and Gina are part of the Airlock team at Genomics England. We have another colleague, Eve, but she's currently on leave, so it'll just be the two of us for today. So we'll take you through how the Airlock system is used to keep participant data and patient data safe and confidential at Genomics England, whilst enabling some great research. I'm going to very briefly go through what Genomics England is. I know a lot of other talks have already mentioned Genomics England, but just to go through a very quick intro, and Gina will explain more later, because it is quite important to give context to what the Airlock system is and what it's actually being used with. So Genomics England was originally set up to deliver the 100,000 Genomes project, which again you've heard referenced during the call, which was of course a program to deliver whole genome sequencing of 100,000 genomes from cancer and rare disease participants and their relatives. And that was for immediate clinical usage, for example, new diagnostic findings, but also to build a genomics and clinical data database for long-term research to be done on it. That's now called the National Genomics Research Library, or the NGRL, which uses data from that original 100K project and from future projects. So yeah, we'll go through how the airlock system is used to keep the data in that database safe and confidential. If I go forward, Gina, can you go to the next slide, please? So what is the airlock? So sadly, we don't get to manage a spaceship component. Gina, next slide, please. So I think the best way to explain the concept of the airlock system is to go through the three kind of broad models of data access that one could have for a biobank of genomics and healthcare data. I think the first thing to mention is just to quickly go through, and again, might be relatively obvious, but I think it's worth speaking explicitly about why it's important to keep genomic data private and safe, or why people might be concerned about that. So of course, someone's genetic data is quite sensitive and private data to them. Someone might very reasonably not want, for example, health insurance companies to know that they carry a certain variant that means that they're predisposed to getting certain conditions, or also that because they have it, their family members also are likely to have it. So there are various reasons someone might want to keep their genetic data private. And there is a dynamic with genetic data for research whereby more detailed descriptions of a participant and more detailed descriptions of their genome can increase the scientific utility of the data, as in richer data sets mean that researchers can do better research because there's more information available to feed into their research. But it can also increase the identification risk of research, the risk that the data could be tied back to a specific participant. So that leads us into now these three broad models of how you could make data accessible to researchers from any kind of genomic data set biobank. So, and they go from the left to right, being relatively more restrictive in terms of how the data is accessible to researchers. So on the left-hand side, we have the idea of an open access data set. That's where the data is available for anyone to view. There's no particular control on the usage of the data. So some cancer data sets like TCGA, the Cancer Genome Atlas, or PCOG, pan-cancer analysis of whole genomes, make a subset of the data available to anyone just on the internet. So you could Google those now, and you could find their websites and download some of the genetic data from the participants who've kindly made their data accessible, the data from their, generally the somatic variant data from their tumors available to those projects. I actually use those for my own master's, so I'm quite familiar with those. But that is, to an extent, the easiest research to work with because it's just freely available. There's no control of what's done with the data, or even tracking, to my understanding, of what's done with the data from those projects. But it's also the least confidential because it's just freely out there in the world. So it might be the relatively more restrictive on what people would be, how detailed data people will be willing to have shown about themselves in context of that data. If we move on then to the middle, we have the idea of a protected access system, which is where approved researchers can see the exact detailed data themselves within a controlled environment, like the genomics research environment, which Gina will be talking you through more in a moment. But there's control on what can be exported and published to the wider world. And this is the idea of an airlock approach, where in our case, we're looking for researchers to be exporting the summarized and anonymized end results of their research rather than the raw data to be shared with the wider world. And Genomics England, of course, uses this approach, as does Our Future Health, as another example of a UK-based biobank with this approach. And again, Gina will be talking more about the details in a moment. And then lastly, on the furthest right side, we have the idea of a black box model, which is where researchers can query the data, but they can't directly see it. And Open Safely is an initiative that allows researchers to query certain NHS data in this way. So they send in a programming script that asks a question, they get back the answer to their question, but they can never see the exact data themselves. And so that is, in a way, keeping the data the most confidential, because even the approved researchers don't see the data themselves directly. But it's also probably the most difficult to work with. Like you'd really need to be quite an expert coder for that to be able to, for you to be able to work with that effectively. And yeah, we take this protected access, like this middle approach, because we feel it's striking the best balance between ensuring that the data is kept confidential and can't be tied back to our participants, but at the same time enabling hopefully the richest amount of research to be done on that data. So I'll pass you on then now to Gina to talk through more of the details of how that works in depth on a day-to-day basis.
Thanks, Pete. So we've already kind of explained the different ways that this research data can be held. So I'm going to show you how this happens at Genomics England, and then give you a little bit more information on our airlock system and just show you what happens with this data on a day-to-day basis. So first I'll explain where the data lives. So this is a diagram showing how this data is fed into the National Genomic Research Library and stored there for the different uses. So the NGRL is a secure cloud-based database that holds de-identified genomic and health data from participants. And it works, I guess, like a secure reading library. So researchers can come in, study the data safely in this virtual library. but they don't take away the raw data. So before anything leaves, it has to come through the airlock system. So I guess that acts almost like a librarian, checking that the results are safe, anonymous, and appropriate to share. So this means that the research can happen while also keeping patients' data safe at all times. So in terms of where the data comes from, a lot of you will already be aware, this comes in through the NHS Genomic Medicine Service, and the rest of the data comes in through previous and ongoing research initiatives, like the 100,000 Genomes Project. And I think some really important context is that when the data is being processed and added to the NGRL, it's first de-identified or pseudonymized. So we remove things like the name and the NHS number of a person, as of course, that information would never actually be relevant to a research question anyway. And all of this data is held by us because the participants have explicitly given us their consent to hold data for this purpose. So now I will move on to what is done with the data and by who. So the data held obviously is in a secure research environment. So to gain access to this, researchers need to be registered with us. So they undergo some checks. So this might be them signing legal agreements. We verify their institutional affiliation, and they also have to undertake some information governance training. So the researchers might be academic or from industry. So it might be anything from PhD students to industry experts. And when they register with us, they also have to present to us the work that they want to carry out so that we can ensure they're using the data for suitable research purposes. And once a research result approved, they can view the data in the NGRL and carry out their research. So obviously complex analytics and bioinformatics to look at the participant data and extract insights. And obviously this might be looking into the cause of a rare disease, or say looking at how a person's genetics might change the way they respond to certain treatments. So Pete's already explained the principles behind having the airlock, and I'll just kind of show you how the data is actually transferred. I'm sure a few of you have used airlock before on this call. So airlock is an application, and researchers can access this from the research environment. And the security of the research environment means that nothing can just come in and out. You can't just copy and paste information. A researcher might request to export the results of their research so they can publish this to the wider world. Sometimes they import data, so this can be obviously used to do further analysis, used as a reference material to compare with the data already held within the NGRL. Practically speaking, researchers submit their data requests to us, just like a drag and drop of the files. We then open the files, assess the content in the context of their research project. If the files pass all of our checks, this will be approved, meaning the files are just sent on to the researcher then and they can access these externally. If the data doesn't pass our checks, we will advise the researcher and work with them to edit their request and make this less identifying. And then we would ask them to resubmit this for a subsequent check and go from there. And of course, in some cases, a researcher might be asking for a new type of data export. We might not have seen this before. So in this scenario, we'll refer this to our airlock committee for discussion, just as an extra layer of governance oversight. So this committee is a panel of Genomics England staff, and they have varying expertise. So areas like clinical genomics, bioinformatics, AI, ethics, data governance. And over time, those decisions made by the committee are used to further build out the airlock rules. And now I will just cover a few of the key principles behind the rules themselves. As I've said, the whole aim of airlock is to ensure that all data transfers comply with ethical standards, data protection regulations, and obviously with the consent that we hold from participants. So all of our rules are created with these principles in mind, and these principles also apply to data from other sources that we end up holding. So when, as I mentioned earlier, researchers might import their own data for comparison, we also will check this and ensure there's sufficient consent for this data to be held within the NGRL as well. And the key is we do want to just strike that balance between protecting the identity of participants while still obviously enabling really valuable research. So at the heart of the rules, we want to prevent the release of any data that could allow participants to be identified. We don't allow individual level descriptions for export unless the risk of re-identification is very low and the scientific value is deemed to be very high. So here on this slide as an example containing made up data, this shows exactly what we mean by individual level descriptions. So in the top pane, this would be rejected. Participant X is 49 years old. They've had an appendectomy in 2016 and they have dyslexia. So this is way too detailed. If you knew this person, you could probably tell who this was about. Whereas you can see in the green, this is a tweaked version, so it's much lower re-identification risk because obviously the age has been put into a range, the disease terms have been broadened out to be more general. So this could be about someone that you know, but it's also broad enough that it could actually apply to quite a few people. And just for context, these examples only show a tiny snippet of what would actually be exported in a request. So this information would be accompanied by something like, this participant X also has this variant, which, for example, is thought to be linked with their condition. So I guess that's where the scientific value element comes in. And just a second key principle that we apply for any requests is data minimization. So if a researcher was doing a project on breast cancer, we wouldn't let them export loads of data on epilepsy, for example. So this ensures that the data released is always proportional to the needs of the given research question, and it stops any data leaving the research environment unnecessarily. And linking to this, we encourage the researchers to analyze their data within the secure research environment wherever possible. So sometimes a researcher might request to export data for further analysis externally. This might be a little bit easier for them, maybe on their own computer. But we'll review whether this work can instead be carried out internally. And if it is possible to do internally, we will reject the request as it would be an unnecessary data export. So this approach also ensures that only summary results such as counts, totals, or patterns are released rather than in any individual level data. So there obviously are lots of different types of data researchers want to explore, and normally this is to form part of research papers, which might then lead to new scientific findings and even changes to clinical practice down the line. And a really nice example to show this is some work that we did to look at the numbers of participants contributing to research studies. So we can look into this because for every ILOC request, we track which participants' data has actually been utilised. And the key takeaway from this was that for the average participant from the 100K, their data has contributed to 137 different projects since 2020. So that could mean their information has helped find answers to 137 different research questions, which is really cool. And flipping that around, so looking at the number of participants whose data has been used to generate one set of research findings. So this project, obviously this was mentioned earlier, this used data from 114,200 people to discover a new genetic cause of disease, so describing a neurodevelopmental disorder which is now named Renew syndrome. And obviously this genetic change was found in a non-coding region of the genome, which is obviously much harder to sequence normally. So I guess it shows how important it is for researchers to be able to access such huge amounts of participant data at once. And I think this was mentioned earlier, changes in this region are now estimated to explain around 0.4% of individuals with neurodevelopmental disorders worldwide, which is a huge amount of people. I think this really just shows the scale of work being done with the genomic data available and how the patient contributions are really feeding into research and bringing about long-awaited diagnoses for families. So I will pass back to Pete now just to close off the presentation.
Thanks so much, Gina. So yeah, that's the full presentation. So I hope everyone found that interesting and useful, and we're very happy to take any questions anyone might have about Genomics England as the ILOC process is implemented or anything around that now. I do see there's a question in the chat about the access that Palantir apparently have or will have to identified patient data. I'm sorry, are we at the Q&A section now, or is it later?
Yeah, absolutely. So yeah, thank you both. If we go through those, I think there's one question for Rachel, and then if we deal with you, we can do your question first, and any other questions, please just pop them in the chat. So there's, how does this excellent secure process sit with the recent debate about access that Palantir apparently have or will have to identified patient data?
Sorry, I thought we were doing the Rachel. No, that's fine. I can answer. Well, or unfortunately, I can answer the question, but with a kind of a non-answer, because the honest answer is I don't know. I don't have any involvement with anything around how Palantir is being involved with patient data nationwide. Yeah, I don't know, to be perfectly honest.
Yeah, that's a fair enough answer. If we don't know.
Yeah, it's all for the future, and yeah, I think it would be separate to, because I do think it is worth mentioning actually. So AirLock sits the interface between researchers who are approved to work on research projects within the Genomics England research environment, and we're the interface that they have to then publish their findings to the wider world, and we're reviewing that to make sure that the data shown is non-identifying. I believe the Palantir interface is to do with how health data is connected nationwide and for active ongoing healthcare, which is kind of a separate thing, although some of that data might be involved in one of the projects that Genomics England runs, and therefore, if those participants consent to having their data included in research, that would then go into the RE. But that's all, I think, separate to what Palantir is going to be involved with. But I don't really know the details, to be perfectly honest. So sorry to not be able to answer it properly. But yeah.
No, that's fine. Thank you. Thank you very much, Peter. I've got one for Rachel, I think.
Yeah.
So do you exclude maternal contamination for the generation study on umbilical cord blood?
When you put the blood through the whole genome sequencer, baby DNA will look different to adult. So yeah, it does. Although that's an interesting question, because you can actually pick up carriers of things. So sometimes it's a bit of a technical answer, but you find that results are in CIS where the baby's a carrier and it doesn't actually have the condition. So this is why we always say that the result is a condition suspected result, because it's a screening test and not a diagnostic result, and they would come in for further diagnostic tests. Does that answer the question? Or
I think so, yeah. Thank you so much. And we've got another one, I think, for the airlock team. So does this include data for family members? Is this consented for? I'm not sure if that's right.
Oh, sorry.
No, so I think it was for you guys.
Okay, yeah. Yes, I can answer this properly. So in the original 100K, family members would have been, so in many cases, family members did, let's say unaffected family members of a participant with a condition, like the parents or the siblings, would have actively signed up as participants. And that's actually why, if you ever hear Genomics England, we have slightly, it might sound slightly insistent terminology, that we say participants rather than patients. There's a few reasons for that, to do it sort of a style guide thing. But one of the reasons is because many of our participants who we have kindly donated their whole genome sequencing or allowed us to host their whole genome sequencing data as a research resource are not themselves patients. They don't have a condition themselves. They are the relatives of someone who has the data. So in the cases where they have, again, kindly allowed us to host their whole genome sequencing data and allowed us to, do that whole genome sequencing in the first place, they would have consented in the same way as a participant who was themselves a patient, themselves a person with cancer or a rare disease. So that would all be in the same way as a patient participant, if you want to put it that way. I don't know if that's our style guide exactly, but they would have consented in the same way. In some cases, family members might have been not contactable or something around there. So in some cases, we do hold a record where it just says this family member exists, like just to be able to say what the family structure is and to say affected or unaffected. So yeah, in those cases, we might have been told by their other family members who did sign up that, well, we have this brother who's not contactable, but they're unaffected by this condition. But that would be the only data we post in them as far as I'm aware. So yeah, I hope that makes sense.
Yeah, thank you so much. Are there any further questions? You can also put hands up and speak. I think I can see hands, hopefully, or you can drop them in the chat. Well, in that case, I've popped a screen up. So we've got a feedback form, which is there to the left. Sorry, that took me longer than it should have done, of the slide. And we really appreciate any feedback that you have, any future ideas or things that you'd like covered. Like I say, we do these on a monthly basis, and we want to try and cover as much as we can in the genomics landscape. We have an excellent network of speakers, as evidenced today. So please do complete that feedback form. And to the right, you'll see all of our platforms. So that's our link tree. It will take you through to all of our kind of upcoming events, all of our socials, all of that commsy stuff that we have. So thank you so much to all of our speakers. We will be, we've recorded this webinar, as you're aware, and we will be sharing that with all registrants after the event, maybe today, maybe on Monday, and we'll send you some good links across, as well as the slides and everything else of relevance. So thank you again. Really appreciate everybody giving up their time today to join us for today's webinar, and we'll hopefully see you at the next one. Thanks very much.
Thank you very much indeed. Bye-bye.
Thanks so much, everyone. Bye.